All right, everyone. Thanks for coming to the last seminar of the semester. Thanks to the ecology lab for the ramen bar. Looks like people are happy, smiling. And then afterwards, we have happy hour. So stick around and chat with our speaker and drink some beer or other beverages. So our speaker today is Mike Fox. And Mike, let's see his background. He did his bachelor's degree in, I forget, somewhere, Florida, San Diego. Was that San Diego? And then he went to Florida. Yeah, why did I keep thinking Florida somewhere? Okay. Oh, well, I knew that now, but okay.
He's from Fresno. He doesn't know.
Fresno. I don't know geography. Okay. And then he comes here. Yes. But did you do diving first in the tropics?
Yeah, I was in the
Yeah. Oh, in the Caribbean. Okay, that's right. That was right. Yeah. So started diving in the tropics. Comes here to Moss Landing. It's like, it's basically the same. I'm gonna go work in these kelp forests. He's in Mike's lab. Jumps in the water. It is cold. He was pretty much shivering the entire time he was here. He was always wearing a beanie and and puffy coat no matter what, because he was always freezing. So Mike did his master's degree, and he was a beer pig, obviously. Worked hard, played hard. He studied the effects of disturbance on giant kelp. So he did a lot of diving and field work. He did a bunch of really cool experiments, going out there and manipulating giant kelp in different ways, cutting it here and there, and then looking at the recovery depending on the type and intensity of disturbance. Mike also went with us down to Chile when we taught the Chile class, and that's where he met up with Seth Newsom, who is an isotope guru, which gave Mike some new skills in his toolkit, and then he also applied those isotopic techniques to his thesis to try to understand how nutrients were flowing and how the disturbance kind of affected the nutrients transfers and stuff in giant kelp. He then decided the cold kelp forests are way too cold. I'm never going to dive in those again, and I don't think he has.
Only a couple times.
Yeah. And then he went and did his PhD, then down at Scripps in Jen Smith's lab, working now on coral reefs. And there, when he was there, he was studying kind of the effects of oceanography and environmental factors on corals and nutrients and kind of what was influencing whether they're acting more heterotrophically or autotrophically. That's, I think, what he's going to be telling us about a lot today. We were able to overlap on a number of cruises, and so he left these cold, dark kelp forests and went and hopped around basically diving on all the most beautiful, pristine places left on the planet. I remember coming out for some of these dives. At the end of the day, we're having a beer looking at each other like, man, we are the luckiest people on the planet. These are the most amazing places, to be able to see and experience. I've also was reflecting back. We've published 2 papers together. One, a book chapter with Mike on kelp as a foundation species, my only kelp paper. And then recently, Mike published with grad student Scott Miller a study on the effects of oceanography on the trophic ecology of coral reef fishes. So he published it on kelp, he published it on fishes, he published it on corals. What can't this man do? I don't know. And now he's doing a postdoc at Woods Hole, kind of following up on a lot of those same issues, studying how oceanography is affecting corals. So, it's great to see one of our Moss Landing graduates come back and be so successful. It gives me a lot of pride in the program that we're making a lot of these, real badass scientists. And so, so great that Mike could come back, and off you go.
Cool. Thanks, Scott. Well, thanks. It's good to be back. Some things don't change. Good. Well, thanks for Cheryl and the IC Lab for making this happen. It's great to come back to Moss Landing. This is definitely one of the highlights of my graduate career, so I hope you guys are all enjoying your time here. What I want to do today is tell you a little bit of a story about reef-building corals to kind of offer you some new perspectives that are emerging from the work that I did for my PhD and I'm expanding on now at Hui. As Scott said, coral reefs are warm, wonderful places to dive, and we've been very fortunate to go to the most remote islands on the planet and try and understand how these systems work in the absence of local human disturbances like overfishing and pollution. And sometimes it's important to remember, other than them being very nice places to dive and work, why coral reefs are so important. So I wanted to drop 3 just kind of big overview points for you. That coral reefs, the structural complexity and the nutrient cycling afforded by these corals causes coral reefs to be home to about 1/4 of all marine biodiversity, despite the fact that they cover a very small proportion of the ocean floor. So they're very dynamic and ecologically interesting places to work. Corals are also the builders of islands and nations in some instances, and they provide the calcium carbonate framework for barrier islands and reef systems that provide people with places to live and protect their coastlines from large storms. And most importantly, corals are the economic engine for millions of people to provide food and revenue through tourism and sustainable fishing. And these are critical ecosystems that are rapidly changing, and as I'm sure you've seen, are threatened by a multitude of factors. But what I think is most interesting about coral reefs is they provide all these really important services for people around the world, and you can trace their ability to do that back to tiny little polyps and the tiny little algae that live inside those polyps, and collectively they build massive structures that can be seen from space. So if you're not familiar with corals, they're symbiotic cnidarians. So this is a Pocillopora colony. This is made up of thousands of genetically identical polyps. Each polyp has a very tight relationship with symbiotic microalgae that are dinoflagellates that share nutrients and carbon with the coral host. And this is the aspect of reef-building corals that most people focus on. And if you look at a coral under a fluorescence microscope, it's very easy to see why. So here are two polyps. The red coloring you see, every little red ball, is an individual algal cell. That's the chlorophyll fluorescing in the animal tissue. And so some species of corals can have up to 3 million algal cells per square centimeter. So it's kind of undeniable that while these are animals, they're also very much plants in some extent. But because of that, the animal part, which is seen here in the green, this is fluorescent protein in the animal pigment, gets kind of overlooked. And that's kind of where I want to focus a lot of the talk on today, is what are corals as animals doing, and what does the other side of their diet look like when they're not relying exclusively on autotrophic nutrition from these symbionts. And the benefits of the symbiosis that corals have with algae are numerous, but it's also kind of their Achilles heel. So I'm sure you've seen much of the news of corals recently as photographs like this in the news. Coral reefs are threatened by global bleaching events and rising ocean temperatures because their symbiosis with those microalgae is very sensitive to small increases in temperature, sometimes as little as just 1 degree Celsius. However, when the corals are white, it's important to know that they're not quite dead yet. They've expelled the algae from their tissues, and so you're looking through the translucent coral at the calcium carbonate skeleton underneath it. But there are still thousands of little hands and mouths on these colonies that are very capable of capturing and consuming prey. So in a situation like this, corals are kind of starving to death unless they have energetic reserves they can draw down or food that they can capture to maintain their metabolic demands. And if corals can maintain their energetic demands long enough and water temperatures cool off, they can regain those algae and continue on their way. Other times they will ostensibly starve, and that's when corals die and start to be covered in that algae. So they look kind of like rocks sometimes. If you swim by, sometimes they're pretty colors. This is a fluorescent Montipora that's just unhappy. And they don't really move a lot. They're not super dynamic. It's not like a shark swimming around on a reef, so a lot of people don't think about corals as predators. So what I want to do is change your perspective a little bit, and I want you to remember this talk next time you're swimming around on coral reefs, because corals are voracious predators. They're a lot like this. They've actually inspired monsters from famous movies. And until you see it, it's kind of hard to fathom. And so I found a lot of awesome YouTube videos that I'd like to share with you. This is a polyp from Acanthastria coral. It's a large polyp coral. And this is mostly in time lapse. It's sped up a little bit, but you'll see that corals are quite dynamic. So an aquarist is going to drop a frozen piece of food here in a second, and the coral rapidly responds, and the food is gone. Okay, so like they're kind of scary looking if they were much bigger. Corals have a variety of feeding mechanisms. So this is another common Indo-Pacific coral, Galexia, and this coral is so hungry it doesn't even take time to ingest its food. So the aquarist is dropping in some brine shrimp here, and what Galexia does is it extends mesenterial filaments outside of the polyp, which are sticky, and they catch lots of prey items at any one time, and the coral will actually digest those and suck in the nutrients and then just spit off the carcasses without ever actually swallowing it. Stylophora is closely related to the coral I'll be talking about in a little bit. It feeds kind of more traditionally, if you will, where each individual polyp kind of grabs and captures one prey item and then ingests it. And these corals can do about 2 prey items per hour per polyp. So their feeding rates, if they have access to food, are pretty high, even though they feed kind of slowly. And then I was recently in Bermuda, and we noticed another cool thing, that certain corals also are very strategic about how they use their tentacles when feeding. So this is a Faviafragum, a golf ball coral, common in the Caribbean. My footage is not quite as nice as those other ones, but basically this is one polyp here. You'll see some activity down here that's kind of interesting, but focus on this. And we have small brine shrimp that are kind of little red blurs in the video. And what you'll see is that this coral maximizes how much food it can take at any one time by keeping tentacles that haven't caught something still active. So it catches the first prey item here and starts to pull that in, but notice that these are still extended. And it knows that there's food in the water because corals have incredible chemosensory abilities. And you'll see that soon another one will swim a little too close to these tentacles and gets caught. But it's got two, and it's still hunting. And actually we see a third one. And this is not sped up. This is all real time. So you can imagine that as corals have their tentacles out on the reef, they can feed quite a bit if there's enough food floating past. So a lot of people have heard that corals only feed at night, and that's true for some species. Where Scott and I work, we're not really allowed to go in the water after dark, or we might not come back. So I can't do much night work. But if you swim around on a reef and you look closely, you'll see that many corals have their tentacles out all day and are actively feeding. So these are a few random photos that I took swimming around. Poecilopora, this is a Fungia, Galexia again that we saw earlier. This is a Hydenoptera coral, Porites. So across genera, across morphologies, across polyp sizes, they're actively hunting. And that's because heterotrophy is really beneficial to corals. It has all sorts of huge benefits. They can consume a wide variety of different prey types, including dissolved organic matter, and they can capture phytoplankton, zooplankton. Some corals can actually consume microbes. And when they get energy from those different sources, they have enhanced tissue growth, enhanced skeletal growth. They build up fat reserves, kind of like we do. They can harbor more symbionts, so they can also become more autotrophic. And their reproductive output goes way up. And the other really important thing about this is that corals, they're sensitive little critters, but if you feed them, they are less sensitive. So you can put a coral in a tank, crank up the temperature. As long as you're feeding it, it's a little more resistant to that heat. It can also survive being bleached much longer. Corals calcify better under low pH if they're being fed. Similarly, negative effects of nutrient pollution can be offset by feeding. So it's kind of this critical aspect of coral biology, and it dictates a lot of corals' energetic budgets, how much they can grow, how many more babies they can produce. But it's really hard to measure, and so we haven't looked at it that closely in recent years, and a lot of the seminal work was done in tanks. And so what we want to do is kind of take this perspective to the field and see if we can learn about how corals are feeding in the environment. To help you get a sense of the benefits I'm talking about, so if this tiny little thing here you can hardly see is a type of coral that was grown by some aquaculturists. And what they did was they gave the corals different feeding amounts and different types, but this is a graph of the size of the coral over a 5-month period. So the blue one was in the ambient, whatever was in the water it was eating, and it was that big at the end of 5 months. Whereas if you feed the coral as much as it can handle, growth is very much nonlinear, and it gets huge. So these are obvious benefits. If you ever talk to an aquarist and you ask them, how do I keep my corals happy, the first thing they'll generally tell you is to feed them. So corals like being fed. They like eating, and it has very noticeable effects. And we learned through some very nice tank studies that the ability of corals to change what they're feeding on and change when they're feeding can really impact how they survive heat stress. And right now on coral reefs, bleaching is one of the biggest, most pervasive problems at a global scale. And the access to food before, during, and after bleaching events can drastically change the outcome of how a coral responds to this heat stress. And so this is something we really need to start getting a better handle on, and we haven't really done it outside of labs yet because, like I mentioned, it's very hard to quantify. And so my motivation for looking into this a little more closely was that corals are bleaching at scales that are somewhat overwhelming. And it's happening more frequently, and we're really noticing significant loss of corals in some places like the Great Barrier Reef, which you probably heard about on the news. So we need to understand if or how feeding is playing into the ability of certain colonies to survive. Like that coral's not bleached, but everything else around it is. there's a lot of genetic work being done to try and assess that, but we can also try and get at, these are mixed trophic organisms. Can they use that to their advantage in some way, and are they able to do that in different places? So to explore food availability and corals' response to that, I spend a lot of time in a very interesting part of the Pacific Ocean. This is Hawaii for reference, where we spend a lot of time is the Line Islands and the Phoenix Islands, which I'll talk about a little bit later. And this here is called the equatorial cold tongue. So that image is showing you surface chlorophyll A concentrations measured from satellite. So generally coral reefs are considered to be warm, nutrient poor, very clear. But as you can see, there are certain areas. This is a blown up graph of a 15-year climatology. This is a lot of the work from Scott Miller's thesis that Scott just talked about. And you can see that these coral reefs, while close together and pretty similar, live in very different places. And primary production has a lot to do with food availability on coral reefs. And so that was something we could actually take advantage of. And some of the data I'll show you today are from Palmyra. Colleagues of ours in Hawaii were also thinking about this, and they recently published a paper looking at a whole bunch of islands across the entire Pacific Basin. And what they found out was not super surprising, but again, we kind of overlooked it, is that the interaction between these big mountains in the middle of the ocean and the surrounding water changes a lot of the circulation and increases things like internal waves and upwelling, or atoll lagoons actually provide all sorts of stuff to the nearshore reefs that we've sort of ignored. So the waters immediately around coral reefs are a lot more productive than classically considered. And that varies as a function of where these islands are in an ocean too. So these reefs have a lot more access to offshore energy than we've thought about. And what I wanted to know is there a way we can quantify that without having to be on these reefs for extended periods of time, because they're hard to get to. And that's where satellites come in. They're really useful. And basically, you can look at the surface ocean and estimate the chlorophyll content. And a bunch of people are very interested in this from a fisheries perspective, or looking at how large animals are tracking feeding patterns and spatial availability of food in the ocean. And what we know is that reliably, measurements of higher chlorophyll A tend to predict planktonic biomass, and that also correlates with the abundance of things that eat plankton or things that rely on other things that eat plankton. And so we know manta rays and other large pelagics can generally follow or occur in higher abundance in these areas. But we also know that corals are attached to the bottom, and anytime there's high productivity water, it's probably washing over them. So what I wanted to know is, can we use remotely sensed climatologies to estimate food availability for corals? And then within that, can we find any response of the corals to increased food? And if you look at a global map of surface chlorophyll A, so more red in the poles is very high, very productive. Blue is sort of the oligotrophic gyres. So these stars are some of the reefs that we've worked on. And you can see that they're not all the same, right? Some, like Hawaii, are quite oligotrophic, or the Marshall Islands over here. But then you have the Galapagos, where Cheryl does some work, and then we have our Line Islands here. And a lot of times the communities are quite similar, but the environment in which they live is very different. And so we want to know, does food availability play any role in how a coral's energy budget works across these places? So I had a bunch of samples from the Line Islands. I worked with a colleague in Australia to get some stuff from the Maldives, and then I dug through the literature to try and find any other measurements I could for corals that came from about 10 meters depth on the forereef. And what I was looking for were carbon-13 values for the corals and their endosymbiont algae measured separately. And I'll tell you about why I wanted that in a second. So coming up with 15 different species across 16 locations, we actually got a really nice gradient in surface production. So this is Flint in the southern Line Islands near Tahiti, Lee Stocking in the Bahamas, all the way over to the more productive reefs of the Maldives, which are influenced by the monsoons every year. And this is a nice threefold gradient. And so indeed, the corals that live on these reefs are not experiencing the same conditions on a day-to-day basis. But I'd like to point out that this is a big gradient for corals, but it doesn't really mean that we're diving on green reefs, okay? The water is still very clear. This isn't a light limitation thing. For contrast, Stillwater Cove, this is me before I decided I didn't like my wetsuit, is 10 to 30 times higher. So we're talking like 3 to 10 micrograms versus less than 0.5. So big and noticeable ecologically speaking. We see that coral reef fish respond to this gradient, but it's not something that fundamentally changes where they live. And the reason I wanted to use carbon-13 values was because Len Muscatine, who was at the forefront of the coral-algal symbiosis back in the 60s, 70s, and 80s, was the first person to try and come up with a way to estimate where coral was getting the carbon in its tissues. And he proposed that by looking at the offset between the symbiont, which has a more negative C13 value because they're carbon limited, versus the host, which can also access phytoplankton carbon, which is more less negative, because they're not carbon limited, that we could get at where the coral was building its tissues from, what carbon was it using. So the way that this would work in a food scenario would be if the corals don't care and they behave the same everywhere, we would expect that this value is flatline. No change in big delta 13C. So corals are probably some version of autotrophic and heterotrophic, but they don't change what they do depending on where they are. If this responded to increased heterotrophy, as we use increasing chlorophyll A as a proxy, we would see that number become more negative as we move up in the chlorophyll A scale. This would imply that corals are incorporating carbon from outside their bodies in waters that are more productive. And so if you synthesize the data from all of those different places, what we found was a surprisingly strong negative relationship between the amount of chlorophyll A over large timescales on average on these islands and how much carbon corals were incorporating in their tissue from outside. So this suggests that corals on more productive reefs are accessing that food and doing something with it that's being recorded in their tissues. So it kind of makes sense, right? You saw how they feed, you've seen what's in the water. And so our working hypothesis is that if you're in an oligotrophic coral reef, much of the upwelling and the food availability tends to be restricted to deeper depths. So we know corals down deep tend to eat more. They're also light limited, but in the shallows, they rely predominantly on their symbionts. On more productive reefs, certain corals that might be good at feeding are capable of accessing that additional input of food from offshore or from a lagoon. And so basically, they have the opportunity and the ability to feed more when there's more food around. And corals aren't the only ones that benefit from this. As I mentioned, Ecology Lab's hosting, so I thought I'd put some fish in. If you swim around on a reef, you'll notice that there are always thousands of planktivores up in the water column, and they're not there just swimming around for fun. Reefs are always being washed over by offshore water, and the amount of plankton that's contributed to reefs is significant, and the drawdown is quite measurable. But we know on more productive reefs, you also see a lot more planktivores, and as Scott Miller worked with Scott has shown that is also reflected in the diets of different fishes. And so corals are sitting right here, and that same water is washing over them. And if they're able to access the food, it doesn't seem overly surprising that they would do that. And this is kind of it's a simple topic, but the coral reef world is kind of just waking back up and thinking about this at a new level. And so these are some titles of papers that have all been published in the last year. There's a common theme, if you note my red underlines here: prevalence of pelagic dependence among predators, pelagic subsidies underpin fish productivity. This is Scott Miller's paper here, expanding niche widths on more productive islands. Upwelling, it's a much bigger source of nitrogen on coral reefs than we ever thought before. It's enhancing net ecosystem calcification in Hawaii. So the productivity of reefs is really an essential mechanism of how these ecosystems work. And we still have a poor understanding of what it does to the foundation species of these ecosystems, the corals that build the reefs and the corals that are the reason that everything else is there. So we need to explore this more. And the problem with using bulk carbon and nitrogen isotopes is that they're pretty limited. And while these graphs look really nice and we have this relationship between potentially more feeding and more productive reefs, we can't say anything about who's eating more than who, how much are they actually eating, what proportion of their energy does this represent. This is kind of an indirect proxy. It's sort of very coarse. And if you try to collect more data and get a better idea of how this is working, what you can wind up with is a mess. So this is a whole bunch of samples I collected on Palmyra where I really thought I would explain this more. Didn't work. Carbon isotope values on the x-axis, your nitrogen isotope values on the y. The brown dots are zooplankton, what the corals could be eating. 95% confidence interval showing you that food in and of itself can be extremely variable in bulk isotopes. And the green dots are the endosymbionts from individual corals. The corals are shown in the blue stars. So you can't do a whole lot with this, and you certainly can't quantify anything. And so what we're running into is that as we really want more precision and we want the ability to say what proportion of a coral's diet is being contributed to by food, we're unable to do that with our current tools, which prohibits us from detecting spatial patterns. We can't, there's limited variation in terms of the carbon number here. These differences are small. Sometimes we run into like instrument precision errors where we actually just can't tease it apart. And therefore we can't define or quantify nutritional contributions using this approach. So because I went to Chile and I met this guy named Seth, I bothered him again. And I went to New Mexico to apply an emerging technique to ask this question in a little more detail. So we need a new tool, and in ecology, the stable isotope analysis of essential amino acids and other amino acids is a very powerful method of tracing energy flow through ecosystems, and I thought that symbiotic corals would be a great test case to apply this new method. This is my colleague Emma running the first ever coral sample through her GC mass spec. And I collected a bunch of a very common coral on Palmyra, Passipora meandrina, along with its food, and we use the symbionts as the autotrophic energy budget. And why this new method is so great is a couple of reasons. First, we get 6 times more data per sample. So if you just run traditional bulk isotopes, you split the algae and the coral, and you get one number for each one. What we can do now is identify and isolate individual compounds. This is an example of the amino acid lysine. And so in each sample, we can split the coral, split the algae out, and then we measure 6 essential amino acids that are here. And essential amino acids are great because higher organisms generally can't produce them, so we incorporate them directly from our diet, which means that their isotope value doesn't change. So in a great white shark, the 13C value of isoleucine should reflect the phytoplankton at the base of that animal's food chain, and they should be identical and traceable through the whole food chain, which is really powerful. We measure a number of other amino acids, but these are the most powerful and easy to understand ones, so this is where we've kind of focused in the beginning. When I was at Moss Landing, I remember thinking about doing this, and it was really expensive, so I made this funny time lapse to show you that doing this is kind of a pain. And it's expensive because it takes 4 days to get samples ready to go into the instrument versus like a couple of hours if you just want to do regular isotopes. So this is day 3 of 4 in terms of what we're doing here, and you sit and chase these things for like 3 hours. And what we're doing is running the samples through, this is a cation exchange resin, and what it's doing is catching all of the amino acids in the sample and letting everything else run through. And then you can change the pH of this column and drop out all your amino acids in the end. So this is a way of purifying and measuring only what we want in the samples, but it's tedious. After this, you then put your samples through 2 high-temperature reactions where things can either explode or disappear. So sometimes you wind up doing several samples more than once. But because we get 6 data points per sample, we can use multivariate statistics to tease apart our food pathways with greater precision. And as I mentioned, essential amino acids aren't modified by the consumer. Their isotope values are fixed by the organisms that make them. And it just so happens that bacteria, microalgae, fungi, terrestrial plants, seaweed, they manufacture these same amino acids in slightly different ways through different biosynthetic pathways, which creates unique multivariate fingerprints for amino acids produced from different organisms. So in some cases, you can actually trace bacterial carbon, fungal carbon, all the way through the food web just by using these multivariate fingerprints. So what we want to know is, can we do this with the coral's dining options, which are pretty simple. It's autotrophic from its symbionts, or it's heterotrophic from its food source. And that would look something like this, where we use linear discriminant analysis on those 6 essential amino acids from the coral symbionts, and ideally they show up in one cluster. And then we compare that fingerprint to the fingerprint of the coral's food, which is fixed by pelagic phytoplankton and the copepods that eat them. And then we could see where the coral falls out on that continuum if we have good separation, and maybe we can start quantifying how much the coral's getting from one source or the other. And that would give us a sort of working framework like this, where we can look at individual level variation in how much carbon they're incorporating from food. Oops, fortunately, it worked. We ran a bunch of endosymbionts. They all clustered out very nicely on the right side of the plot. And food sources, triangle and square. So triangle is POM, or phytoplankton. The hypothesis is that those values should be identical in the copepods that are eating them, and you see that they all cluster together. And what was awesome is that we had very strong separation between these two food sources. One of the ways you quantify how good your separation is you take a dot out and ask the statistics to classify it as one of your groups, and usually 80% and above is good, and we get 100 every time for each group. So these fingerprints are very unique, which is great. And what you can do now is place the corals into this mixing space, and the hypothesis with mixotrophic corals is that probably a bunch of them are over here. Maybe we're going to get some in the middle. We didn't really know what this was going to look like, and what we found was a little surprising. Our corals fell completely across the spectrum, and we had about 34% of the Poecilophora colonies we sampled were getting more of their amino acids from heterotrophy than they were from autotrophy. And we had 98% of the variation explained in our x-axis, which is very convenient, because now we can basically just think about coral feeding as a left-to-right process. And that allowed us to do some resampling to estimate proportional contributions of heterotrophic nutrition to the diet of individual Poecilophora colonies. So this graph shows that. So each bar is an individual, and this is the percent of heterotrophy in their diet from 0 to 100. And you can see that we have this crazy continuum of vegan corals down here that appear not to be eating anything, and other corals up here that something's going on, they're eating a lot. But on average, Poecilophora was getting about 40% of its essential amino acids from heterotrophy. And you would think that maybe these corals are all in the same place, and they live in a weird spot. There's more food. Maybe they live underneath a rock or an overhang. But if you break them down by where I got them, what we find is that actually it's individuals who are doing different things. And at each of these 4 sites, you had a coral eating a lot and a coral not at all. And so that is a little confusing because corals are considered to all sort of behave the same. But we know from a lot of the genetic work that genotypes is a very strong and important variable constraining physiological responses in corals, and so possibly their ability or their preference for feeding is also something constrained at a colony level. So when you're swimming around on a reef, looking at something like this, it becomes a lot more complicated, because now you're wondering, okay, well, who's doing what and why? Is it a coral back here that's eating more, or something down here? So we have no idea. But I think now we have the tool that we can start to ask some of those very basic questions and get better answers and understand how this assemblage of corals is partitioning resources or changing their physiology and their metabolic demands to suit their needs. So that's kind of where we're going. We want to apply this new tool to ask very basic questions about how corals are accessing food. We also know now that individuals are a more complicated story than we thought, but we also can't rank species above one another. Do all plating corals rely primarily on autotrophy? Are these big polyp lobophilias really hungry? We can now define that space with this new technique, but we've also learned that coral reefs are incredibly dynamic physically. And the dynamics of the water directly control food supply, heat stress, all that stuff. And so really we need to assess feeding and food supply and individualness in the context of the physical environment, which can be highly variable around different sides of the same island, even a kilometer down the reef. So this graphic here is from some work we did on Palmyra, looking at how internal wave frequency was changing food supply to corals around Palmyra. So this is the reef slope, which we found to be an incredibly important determinant of how far up a reef internal waves will propagate. So now you have to think about where your coral is and what the reef slope is like relative to another place. And these arrows show the direction and magnitude of the water, and the background color is going to be temperature. So you see we're working on almost a 2 degree, about 1.5 degree temperature scale here. And this is over the span of like a couple of hours on one day. So remember, coral reefs are warm, oligotrophic places where nothing interesting happens. So if you look at 30 meters, you see an internal wave hits the reef slope of Palmyra, and all of a sudden you have this cold water bouncing up the reef slope and coming all the way up to 10 meters. And we know from a lot of work in the Florida Keys by Jim Lecter, who's been at Scripps for a long time, probably taught Mike a class or two, that these internal waves bring up a lot of plankton and a lot of nutrients. And so if you're a coral and your reef is very steep, many times those internal waves just bounce off and never make it to you. But if your reef slope is very gradual, they can come up and be entrained and stick around for quite some time. So now we're trying to look at how, within an island, what does your internal wave space look like, and how does food supply and trophic response of fishes and corals vary within islands and across islands. And at Hui, I'm working with hydrodynamic modelers to look at a slightly larger scale dynamics of flow patterns around individual islands. So this is Canton Island in the Phoenix Islands. I'll show you where that is in a second. And this is during the huge bleaching event in 2015. So this is just going to show you that these are incredibly dynamic systems, and that your heating patterns and your flow patterns are very different depending on where you are. So these are daily cycles on the tides coming into the lagoon, out of the lagoon. But you can see that different places around the island are experiencing very different temperature conditions. Obviously, the lagoon is getting a lot hotter, but this corner of the island has avoided a fair bit of the heating. And so we also know that a lot of food comes out of lagoons, so depending on where that plume goes, corals here may have more access to resources that other corals don't. And so if we can model what this looks like, then we can devise sampling strategies to try and encompass some of this variability before we even show up. And we can explore patterns of survival and ecology and benthic structure within the context of the physical environment in which these organisms live. And what's cool is that what we're trying to find is how the differences in food supply and hydrodynamics and water flow are causing patchy survival. So when a heat wave comes to an island, sometimes you can lose 80% of your corals, but it's not uniformly distributed across the reef. And this is a photo from Canton, where mortality was about 40 to 60% on average around the island, and then sort of accidentally, we dropped on this site because I was looking for something, and it wasn't one of the long-term monitoring sites, which we usually visit when we go to these islands, and we jumped in the water here, and it was like nothing had ever happened. But you can use skeletal cores from long-lived massive corals like this to figure out if the corals at this site did bleach in 2015, and they did. So we know that it got hot enough to bleach corals here, but for whatever reason, survival was a heck of a lot higher. So we're trying to understand the mechanisms that allow that to happen. And the long-term goal of all of this, and the reason I'm at Woods Hole, is because productivity around the tropics changes over long time scales and short time scales. One of the big ones is El Nino, which causes a lot of the heat waves. And there's been some phenomenal work done on skeletal cores of corals to reconstruct sea level rise, rainfall patterns, bleaching histories, changes in nitrate or nitrogen deposition from the atmosphere. So there's a ton of information in coral cores. And actually, there's a lot of organic material locked into the skeleton that we've only recently sort of figured out how to access. And the amino acid profile of the coral skeleton is identical to the coral tissue. We don't know if there's a weird fractionation effect or whatever, but we do know that there are the same amino acids we've been measuring in the tissue locked in that. So what we're trying to figure out is how to get them out and run this sort of same analysis to look at patterns of food supply and bleaching response and growth over long timescales, which could be really cool. And that's a lot harder than I thought it was going to be, but we'll get there. So I wanted to show you really quickly some of the latest data that we're coming up with. And so one of the key things to have this be relevant, so going back in time, we're not going to be able to find like archived phytoplankton from the 1982 El Nino event. But if the fingerprints of coral food and the coral symbionts are consistent across space and across time, then we can try to sample the coral downtime and then place those values in this framework of autotrophy and heterotrophy that we think is pretty well constrained. So the first step to figure out if that's even worth trying is to go to more islands and not just make these measurements on Palmyra. So a lot of my postdoc work is focused here in the Phoenix Islands. So Hawaii is about 1,000 miles up here north of Palmyra. American Samoa, Tahiti's over here. So we sailed through Christmas Island and then Canton. Oh, I think I boxed them, yeah. So these three islands I have data from now. I also have samples from Arona and Rawaki. And the first step is to see if there's any consistency in the isotopic, the multivariate fingerprints of food and symbionts across the space, and then to place corals from those islands in that, but I don't have the coral data yet. So in our multivariate world, if you remember those other graphs, food's on the left, autotrophies on the right. This is the data from Palmyra. The ellipses I'm going to show you represent the 95% confidence intervals of the data points that we have. Palmyra is our best sampled data set to date, so these are pretty tightly constrained. You'll see some funny shapes as I show you other data. That's just because there aren't that many points. And remember, this was from 2015, okay, in Palmyra, which is about 1,000 miles north and a little bit east across the upwelling zone on the equator from Palmyra, so a fairly different water mass in a completely different hemisphere. And I visited them 3 years later. So when I plotted data for the food and the endosymbionts from Canton and the Phoenix Islands, it was encouraging that we saw a lot of fidelity here. The cool thing is that this is a different species of coral. So Postlepora on Palmyra, these are Porites endosymbionts, probably similar endosymbionts, but from 2 different species nonetheless, and we don't really see them changing. We went to Canton's lagoon and grabbed more phytoplankton. They look the same. And Nicomaruro, which is the most oligotrophic of all of these islands, and then at least 1,500 miles away from Palmyra. And again, we see a lot of continuity here. So the fact that these fingerprints are, and there's 93% of the variation is again explained on our single axis. So we think that this is going to be a really nice and useful framework for trying to explore heterotrophy versus autotrophy in different species of corals from different places. And that's kind of what I'm focusing on now. So as you move towards the beer, corals feed more on reefs that have more food. That's kind of our working hypothesis. And so far, it seems that not only are they feeding where there's more food, but certain individuals are feeding more than others. Food availability is highly variable within reefs and among reefs, so not all coral reefs are the same. I think it's really important that we start considering the oceanographic context in which these ecosystems are living to understand the total energy budget for these systems and where that food and those resources are going. Right now is a great time to kind of be asking these questions because we have emerging compound-specific techniques that are transforming our ability to tease apart small signals. And the remote sensing technology is awesome, and it's been around for just over 15 years now, so we have a long enough data set that we can quantify the climatologies of places and get a sense of what their seasonal patterns of productivity are. Are they more productive than other places over the long term? So that data is becoming a lot more useful now than it was 10 years ago. And why we want to do this is because feeding might play a big role in corals surviving various stress events. And if that's true, then understanding where corals have access to food is going to help us understand where they might have a better chance of surviving or where reproductive output is going to be key to come from and repopulate reefs after big heat events. And so hopefully thinking about reefs from a productivity food supply perspective might give us new insights on targeting areas that have higher likelihoods of survival or maintaining diversity through time. And so that's kind of where the next few years will take me. But that's, a lot of people have helped. I've had some great support from multiple institutions. Many of those connections were established here at Moss Landing. So it was after Scott got the job here when we were on Catalina Island. He's like, what are you going to do next? I was like, I don't know. I want to go back to coral reefs. He's like, you should probably email my friend Jen. You guys will get along. We invited Jen here to give a seminar, and now I'm here giving a seminar because I went to Jen's lab and did all this work. So this is a great place to take advantage of the connections you make. And if you have any questions, thanks. Yep.
So if you satiate Nigerians with food, do they need their endosymbionts?
They can live in the dark without symbionts as long as you're feeding them.
Okay, so if that's the case, and then you go to a place like the Mendocino coast or the west end of Galapagos at like Isabella or Fernandina, where you have persistent year-round high-intensity upwelling, would you expect to see endosymbionts in those Nigerians? And do you see a pattern with persistently high productivity? Because these are pretty wimpy productivity values, right? Corals live in higher productivity values. And then under those cases, do you see a trend with endosymbiont, with relying on endosymbionts? Or, I mean, is there a cost to having an endosymbiont where if you're getting enough food, you wouldn't bring them in at all?
With reef-building corals, the symbiosis is not optional for the most part. I mean, it's kind of key to their overall waste excretion, calcification. So I don't think there'd be an instance in which they would willingly get rid of them. In other cnidarians, like there's a coral on the east coast that some colonies living right next to each other choose to have or not have symbionts. It's a facultative symbiosis. So in those, under more productive environments, I would imagine you see fewer relying on autotrophy. In Woods Hole, it's not a seasonal thing either. Productivity is and temperature is, but the colonies of this one coral that have endosymbionts tend to keep them all the time, which we don't really know why. So we're actually doing a case study on those with this method to see if it pans out on corals we know aren't photosynthesizing. What I do expect is the relative proportion of heterotrophy to increase to corals that have access to more food in the coral species that can access it. The one thing that's tricky, though, is, and Cheryl might have some input here, in places like Panama and the Galapagos, and in some instances over in like on Dongsha, when you have huge internal waves or consistent upwelling, the temperature drops. So there have been observations that under extreme upwelling in Panama, corals kind of shut down, and they're not feeding anymore because it's too cold. The hypothesis is that if you increase food supply but drop temperature too much, then those benefits might not be realized because the corals are too busy shivering. So there's a trade-off in like a Goldilocks zone there somewhere, but we don't know what it is.
Fantastic talk, thank you. So my question, you see a lot of variation within a site in terms of the number of colonies that are heterotrophic. Have you looked within a colony? Because oftentimes you'll have clones, right, of the same colony in the same area. And so I'm curious if you see within colony variation as well.
Great question. No, not yet. I have collected some samples for that, but we haven't done anything with them yet. But I would imagine, and some people have done with bulk isotopes looking at top and side of colony, and they do see differences which are driven by light environments. And corals can actually shuffle their symbionts or keep different symbionts on top and side. So we know that intercolony variation is a big thing, and it's pretty dynamic. With what I do, I try to standardize all the branches I take from like top center to try and avoid some of that. And a lot of the colonies we collect are not, I make sure they're not like down in a hole or something. But I would expect intercolony variation to be pretty high. But what we don't know is like, if you sampled 5 branches and averaged it out, do you get the same value or something totally different? But that would, it's something we totally need to do. Yeah?
The systems I work in are high islands that have a lot of freshwater runoff, which can kill the corals. Yep. But actually that freshwater can often be really full of nutrients. Mm-hmm. And so I'm really curious if, and it can also bring in pollutants and other things which aren't good for the corals, but I'm curious if you guys have looked at any terrestrial subsidies, nutrient subsidies?
The short answer is no. That's a really good question. Yeah, the like terrestrial subsidies are, it would be tricky. I imagine like the primary productivity stimulated by some of those runoff events could come in, but I think there's so many other variables that we'd have to take into account. Like you and I were talking about this last night, that sometimes the chemistry of that water that's coming out of those places or out of groundwater injection sites is just so bizarre that the corals might be doing very different things just because their environments change. So I think isolating those would be tough. But there's a lot of work going on in Hawaii right now looking at wastewater injection sites and groundwater seepage on nutrient budgets and coral calcification. And so if that stimulated food and the residence time was long enough that it stuck around and didn't compromise the coral in other ways, I would imagine they'd respond. There's one paper that claims to have evidence of corals eating seagrass, which is probably the closest thing I can think of to something like that, where there's a particulate carbon coming in from outside the coral reef that the coral is accessing. And the same would probably be true from like mangrove habitats and stuff. And we know corals thrive in some of those weird places, but we don't know what they're consuming. Yui.
Yeah, awesome talk. So you were able to, by using amino acids, you can look at the proportion of nitrogen, right, from mixotrophy versus autotrophy. are there similar techniques that you can get like percent power budget? So like what the energy, the joules that come, the energy the organism expends, and then could that explain, like if you look at the metabolism patterns of reefs worldwide, some are autotrophic, some are heterotrophic, but I'm wondering if you can validate that with another independent approach and kind of get at why.
Yeah, that's really interesting. The closest thing I've seen to that, I think we could do it. We'd have to do a bunch of other measurements to kind of parameterize how that would work. But a number of the laboratory feeding studies have, measured photosynthesis and respiration and determined energetic demands, and then looked at the carbon content of food and measured feeding rates and stuff. And they've come up with these. It's CHAR, contribution of heterotrophy to animal respiration, is kind of the metabolic metric that's used. We can probably do something like that with finer precision, but we'd have to do a bunch of experiments to see how that scales on a coral we can't measure the feeding rate with. But I think your point of understanding the overall ecosystem function, I think, is a critical piece of this, because the corals are behaving differently if they have access to food, right? Like the picture of the one that grew 10 times more, like your calcification budget on a reef full of those guys is going to be very different than corals that aren't growing. So I imagine they're pretty interrelated. Yeah.
But what about the fish? Fish out here. Well, you and I were talking about nutrients for fish. Like, I don't know if Jake has worked in that area. But what about those nutrients from those fishes? And you're talking about spatial variability differences. I feel like if he has for those species done, it would be fairly easy to look at that as a source of nutrients, because you're looking so much about slope, gradient, and upwelling derived nutrients.
Yeah. So, yes. And they actually showed that certain large branching acropods in the Caribbean, where you have a bunch of grunts that kind of hang out under them during the day, can have enhanced growth just because the fish are peeing on the coral all day. And in a number of laboratory studies, they've used nitrate or ammonium to simulate natural fish pee, and generally that's less harmful to corals. It messes with the symbiosis less. So we know, and they've started trying to quantify it, and I think similar to what you're finding is that the amount of nitrogen fish put onto a reef is huge. But that's also kind of just an emerging thing in coral reefs, and I think Darren Brickhoff at Santa Barbara is actively working on that now. I don't know. I think that would be a much more direct nitrogen uptake thing for the coral. And I have some work from Hawaii looking at nitrogen uptake in different species, and two species can have fundamentally different responses to increased nutrients. So we also don't know who's responding or not. But a crab living in a Pocillopora colony, or the hawkfish that sit on branches of certain Pocillopora colonies or not, could totally have access to a different nutrient budget than a coral sitting right next to them that doesn't. So I'm sure it further complicates this. Yeah.
Look at all the other things you got to measure now.
That's why we have you. You can do so many things. Yeah.
I was also just wondering, I kind of remember from Scott Miller's thesis, when you're looking across the productivity gradient, are some of those islands fish versus not? I couldn't remember. So they're all not fish.
They're really hard to get to. There's no good fishing port close, so they're sort of isolated by geography, which is nice.
I guess a couple of people Christmas.
Oh yeah, but he
Not
was
the sevens, not the sevens.
Yeah.
in
So
the northern.
the full northern, there's 3 that are active.
Great.
That are fish, keeping them there.
Your gradient you were looking at was just the southern line items though?
It spans the whole thing, but Scott Miller's work and my work was focused on southern plus Palmyra, and now I'm going with Christmas and Jarvis and trying to fill it in. But the islands, the 11 islands span a huge gradient right through the equator. And in the southern islands where Scott's work was, the 2 northernmost ones are at almost 4 degrees south, so it's pretty close to the equator, and then Flint, which is very nutrient poor, is about 12 degrees south. So there's a strong gradient just in those islands too. And we see inorganic nitrate go from about 5 to less than 1. So on a reef, a lot of when people first started reporting the inorganic nitrate values on these reefs in the Central Atlantic or Pacific, people just assumed we measured on them, or we contaminated samples. On Jarvis, which is right on the equator, you can have micromolar nitrate of in excess of 14, which is almost like what you're seeing with silica. That's their really strong components. So they're, dynamic places. But they also have some of the highest fish biomass and health up until 2015, some of the healthiest reefs on the planet, despite the fact that the nutrient levels are 5 to 10 times above what people think corals can survive in. So that what the nutrients are doing there is a big story that people are working on. And I would imagine it's further complicated by the fact that there's way more fish on those islands too, eating more diverse diets.
Right, one more, and then we keep talking, it's right here.
You may have addressed this already, but when you were looking at your isotopic signatures, did you have trouble teasing apart the autotroph signature from, like, did you see that showing up in the zooplankton from them eating the phytoplankton, and then that was kind of,
Sharing it, you mean?
yeah.
We thought about it, we worried about it, but the distinct difference in those fingerprints suggests that it's not a huge influence there. And because we're measuring specific molecules, it's unlike, because the coral's dinoflagellates can manufacture those essential amino acids, so the coral wouldn't really give it an essential amino acid from feeding. Probably not. I mean, we don't know, but we would assume that doesn't really make too much sense. So because of the molecules we're measuring, that problem is a little bit less. But in bulk isotopes, what you're doing is you're just trying to rip apart the animal and the plant, and you sit at a centrifuge and you just do it repeatedly. So you hope you don't have any contamination of one half or the other, and in that instance, it's a much bigger concern, especially because the differences in those numbers is really small. But I think we can isolate that problem by measuring the essential amino acids instead.
All right.
Cool.
Thank
Thanks.
you, speaker.